Hello children! The living organisms survive under different conditions and acquire food in different ways. We know about autotrophic nutrition in plants through photosynthesis. The organisms that cannot make their own food are heterotrophs. Let's know about heterotrophic nutrition now. I am Bindu. Will explain to you about heterotrophic nutrition from the lesson Life Processes of Class 10th Biology. Learning Outcomes. After studying this topic, you will be knowing about nutrition process in amoeba and paramecium, parasitic nutrition in cuscuta, nutrition in humans. Keywords: heterotrophic, parasitic, Hostoria, alimentary canal, salivary glands, peristaltic movement, sphincter, villi, emulsification. Let's see the concept map on heterotrophic nutrition. How do organisms obtain their nutrition? Depending on the type and availability of food, organisms have different strategies for food intake. Some organisms break down the food materials outside the body and then absorb it. These are saprophytes like bread molds, yeast, mushrooms, etc. Some other organisms derive nutrition from plants and animals without killing them. These are parasites like cuscuta, lice, leeches, and tapeworms. Others take in the whole material and break it down inside their bodies. Since different types of foods are obtained in different ways, the digestive system is also different in various organisms. You are aware that amoeba is a unicellular organism. All life activities must take place in the same cell, and the food may be taken in by the entire surface. But as the complexity of the organism increases, different parts become specialized to perform different functions. The amoeba takes in food using temporary finger-like extensions or pseudopodia of the cell surface, which fuse over the food particle, forming a food vacuole. Inside the food vacuole, complex substances are broken down into simpler ones, then diffuse into the cytoplasm. The remaining undigested material is removed to the surface of the cell and thrown out. Paramecium is also a unicellular organism whose cell has a definite shape. Food is taken in at a specific spot. Food is moved to the spot or cytostome by the movement of cilia, which covers the entire surface of the cell, where the food is ingested. Parasitic nutrition in Cuscuta. Dodder or Cuscuta is a leafless, twining, parasitic plant that belongs to the morning glory family. Convolvulaceae. It has no chlorophyll and it absorbs food through haustoria. These are root-like structures that penetrate the tissue of a host plant and may kill it. The slender, string-like stems of the dodder may be yellow, orange, pink or brown, and the leaves are reduced to minute scales. The dodder's flowers, formed in nodules-like clutches and contain yellow or white bell-like petals. The dodder seed germinates, forming an anchoring root, and then sends up a slender stem that grows in a spiral fashion until it reaches a host plant. It then twines around the stem of the host plant and forms haustoria, which penetrates through it. Water is drawn through the haustoria from the host plant's xylem, and nutrients are drawn from its phloem. It spirals around the stems, forming new haustoria, and envelops the host plants as a densely tangled web. Nutrition in human beings. The human digestive system is much more complex. Different parts are involved and perform different functions by using various digestive juices and enzymes. The alimentary canal is a long tube extending from mouth to anus, and various parts of the system perform various functions. Let's observe the flow chart of the human digestive system. Food is cut and crushed by teeth in the mouth and mixed with saliva to make it a wet and slippery form called a bolus. This process is also called as mastication. Saliva is secreted by 3 pairs of salivary glands. 2 pairs are located at the side of the jaw and below the tongue, and a pair is located in the palate. Saliva contains an enzyme amylase or ptyalin, which helps in the breakdown of complex carbohydrates into simple ones. The tongue helps in mixing the food and pushing it into the next part. The lower jaw also helps in this entire process. The soft food mixed with saliva passes through the esophagus by wave-like movements called the peristaltic movement to the stomach. In the stomach, food gets churned with gastric juice and HCl. Now, the food is in semi-solid condition. The digestion of food goes on as most proteins are broken down into smaller molecules by the enzyme pepsin. Food in the form of a soft, slimy substance, partially broken down proteins and carbohydrates is called chyme. Now, the food material passes from the stomach to the small intestine. Here, the ring-like muscles called pyloric sphincter relax to open the passage into the small intestine. The sphincters regulate the passage of only small quantities of food material into the small intestine at a time. The small intestine is the longest part of the alimentary canal. It is the site of further digestion of carbohydrates, proteins, and fats. It receives the secretion of liver and pancreas for this purpose. These juices render the internal condition of the intestine gradually to a basic or alkaline one. Fats are digested by converting them into small globule-like forms with the help of the bile juice secreted from the liver. This process is called emulsification. Pancreatic juice secreted from the pancreas contains enzymes like trypsin for carrying on the process of digestion of proteins and lipase for fats. Walls of the small intestine secrete intestinal juice that breaks down the small molecules of proteins into further smaller molecules. The same is the condition with fats. Carbohydrate digestion started in the mouth and did not occur in the stomach. It resumes now as the medium gradually changes to an alkaline one, and the enzymes become active for carbohydrate breakdown. Let us study the chart showing different enzymes and digestive juices and their functions. Transport of the products of digestion through the wall of the intestine into the blood is called absorption. Internally, the intestinal wall has several finger-like projections called villi. The villi increase the surface area for absorption. Blood vessels and lymph vessels are present in the form of a network in the villi. Products of digestion are absorbed first into the villi and from there into the blood vessels and lymph vessels. Thus, after maximum absorption of food in the small intestine, the rest passes into the large intestine. Here, most of the water present is taken up from this material. This material is then expelled through the anus, which is the last part of the alimentary canal. This passage of undigested material from the body by way of the anus is called defecation. Food that passes out of the anus still contains many proteins, fats and carbohydrates, roughages and fibers of either carbohydrates or proteins. Points to remember. The animals are heterotrophs. Amoeba is unicellular and extends pseudopodia to capture food. The human digestive system is much more complex. The large complex food molecules such as carbohydrates, proteins, lipids, etc. are broken down into simple molecules before they are absorbed and utilized by the animals. This process of breaking down complex molecules into simple molecules is called digestion. In human beings, the food eaten is broken down in various steps with the help of enzymes secreted by digestive glands, which are associated with the alimentary canal, and the digested food is absorbed in the small intestine to be sent to all cells in the body. Home assignment.